Through innovation, academic excellence, and family-centered clinical care, Children's Mercy Kansas City is transforming outcomes for children around the world. Welcome to the audio interview series, Transformational Pediatrics, with host Dr. Michael Smith.
Our topic today is Cancer Genomics, finding answers through data and DNA. My guest is Dr. Erin Guest. Dr. Guest is a pediatric hematologist, oncologist, and director of the Cancer Genomics Program and Cancer Center Biorepository at Children's Mercy Kansas City. Dr. Guest, welcome to the show.
Thank you so much. Glad to be here.
So, let's just maybe step back for a moment because so much is happening in the field of genomics, right? Especially cancer genomics. As you as an expert, I kind of want to just get a nice summary. Where do we stand today really in understanding genomics and pediatric cancer?
We've come a long way in the past 5 years. It's a really exciting field because it's really helping us to understand not just how to learn how to better treat the diseases, but really how to better categorize. So we can take genomic information and we can make subcategories of diseases that previously were described by the way they look under the microscope or some other individual genes that might be mutated in a cancer. But now we can learn so much more about the origin of where the cancer may have come from, and in some cases, we can help families understand why their child developed cancer in the first place, and that's another really important aspect for pediatric cancer, especially when most of these diseases are not being caused or are potentially being caused by genetic changes rather than environmental influences.
Yeah, so Dr. Guest, you kind of alluded to this. Obviously, genomics is a huge field, right? I mean, there's so many different things to study. So what exactly is the focus at Children's Mercy, and what are you really hoping to accomplish?
So we're really, really fortunate at Children's Mercy. We have a Center for Pediatric Genomic Medicine where we have the latest state-of-the-art sequencing equipment. We have our own in-house pipeline informaticist. We have a molecular oncologist who is helping us with this type of work. And our focus has been on helping with diagnostics, helping, like we mentioned, helping determine what might have caused the cancer in a patient if they might have a predisposition syndrome, but then taking a really well-rounded look at the cancers. So not just looking at the DNA in the cancer itself, but the DNA is really just the tip of the iceberg. And we're interested in, of course, what those genetic mutations are, but we can use sequencing technology now to look at the RNA, which is what's being expressed by the cell, and what's being translated then into protein within the cell. So that kind of helps us really figure out what makes a cancer cell tick. And then we can also use some of the really, really cool new technologies of single-cell sequencing, where we can look at just one individual cell, and we can see what the mutations are in that cell and how that cell is functioning and what RNA is being expressed. And we can look across an entire tumor, and it's really interesting that not every single cancer cell in one tumor is identical to each other. And in a patient that has a tumor with metastatic disease, the original tumor and the metastatic disease may be different, and we can use that single-cell technology to really hone in on how has the cancer changed over time, how is it using genetic mutations and changes to get around the treatment that we're providing, and then how can we use that to our advantage to improve our treatments.
To me, that the whole thing that's so fascinating to me, Dr. Guest, is the idea that so this cancer has a certain genetic code, but then understanding, right, what influences the expression of some of these cancer genes and some of these mutations, that could be very powerful, right? I mean, that's I think what people kind of refer to as kind of the epigenetics, the above the gene influences on the expression of these mutations. I mean, what do you think about that? That to me seems like such a powerful thing to be studying.
Right, exactly. And our new our new Genome Center director, Dr. Tomi Pastanen, comes to us with a background in epigenetics, and so that we've really been able to start to incorporate that into our cancer research as well. And I think that's going to really influence how we think of cancers. So a cancer may or may not have a genetic mutation, but if there are changes in the methylation marks on the DNA, that can also change how DNA is expressed. And so we can no longer just think of it as mutations in isolation. If we do that, then we'd miss a lot of that information that helps us understand what genes are turned off, what genes are turned on, how is that changing over time, how does that change as a result of treatment, or as that cancer sort of learns how to work its way around the treatment, especially if it's not responding very well. That those methylation marks are really important. So we can look at those in our lab. The way that we do that is a methodology called whole genome by sulfite sequencing, where we can look at the entire genome. We can look at those methylation marks that are tagged onto that DNA. So that's another area of brand new technology and research that I think is going to impact how we actually treat cancer very, very soon.
Right. And stay ahead of the cancer during that treatment as well, right? So as you mentioned, the cancers are able to resist some of that treatment as they start to learn how to get around that. And then by understanding the genetics and the epigenetics, Dr. Guest, you're kind of staying ahead of that a little bit, and you can anticipate what some of these cancers might do and be ready to offer a different type of treatment. It really is fascinating. But I think a lot of physicians today, and I think a lot of lay people today, still think of the genomics, the cancer genomics, as something in the future. And there definitely is a lot of research we still need to do, and we understand that. But aren't there things going on like right now today in cancer genomics that are helping patients live longer?
Sure. And we are using the technology to reclassify diseases. We can do, for an example, we can do testing of a gene called BRAF or BRAF. And if we find a mutation in that gene, that can actually change how we might approach the treatment for that patient. So that's a test that we regularly do in our lab. And there are a lot of other examples of tests like that. But what we have really felt like we can't just focus on the genes that are known, because there are going to be so many more things out there to discover that are going to impact how we think about cancer and treat it. So what we're doing at Children's Mercy is doing clinical testing, but at the same time using research funding. We're very blessed to have research funding from a lot of different community organizations, from Black and Veatch, from Big Slick, from Children's Mercy Cancer Center, and now our new Children's Research Institute. And all these funds, we're putting them toward being able to do innovative research investigations for all of our patients with new diagnoses of cancer and with relapses, so that we can start to put all of those pieces of the puzzle together, and then all of that research in combination with research being done around the country and around the world is really going to help us advance the treatment for all children with cancer.
Yeah, it does. To me, it seems that Children's Mercy is really in a great position, right, to be a leader and an innovator in cancer genomics. I think that's very exciting for Children's Mercy. But so let's just, where do you see all of this going? And so it's a fascinating field. It's a big field. We touched on just a little bit of it. And what excites you most about this, and where do you see this going?
I think that the things that really get me excited are particularly the single-cell sequencing, and then looking at the RNA. So those sort of newer technologies that are going to go beyond just what the mutations are, and the methylation as well. So I think that I imagine that in another 5 years, we're going to be doing clinical testing that's going to focus on looking at individual cancer cells. So right now, when we take a look at a tumor, at least the standard way of doing that is to kind of mash up all those tumor cells, take all the DNA out of all of the tumor cells all at once. But if we can really understand what's going on in each cell and how that changes over a course of months for a patient's cancer, and if we can get down to the single cell level where we can just do a blood test, and to be able to, maybe a patient has a tumor in a leg bone, but if we can draw their blood and find the information that we need out of that test on an individual cell level, that's going to cut back on a lot of invasive type procedures for patients, and make such a huge difference for their care.
Well, just such an exciting field. Dr. Guest, I think you're just going to have to come on the show occasionally, right, and just give us an update of where everything is at and where we're going, because it really is such a huge field. And I know Children's Mercy is a leader, and there's so much research going on, and I'm excited for you. And I also just want to thank you for coming on the show today. You're listening to Transformational Pediatrics of Children's Mercy, Kansas City. For more information, you go to childrensmercy.org. That's childrensmercy.org. I'm Dr. Mike Smith. Thanks for listening.